the DNA database lab. So we start with the database, and what we're going to do is we're going to type in name of a protein that we're interested in. So this is the hemoglobin beta subunit. And what's going to do is going to give me a list, and it's just giving me a list of the other hemoglobin sequences. Um, there's an entry number for each sequence, as well as the name of the protein and gene, as well as the organism that it originates from. So what I'm going to do is I'm going to click on one of the entries, and you notice a green bar it gets created down here at the bottom, and notice it gives me the entry number that I've selected. And let's say if I add more sequences that I want to compare, I'm just going to pick just sort of 6 here. See, it adds those entry numbers down here. And let's say I want to compare these, compare each of them to each other. First I'm going to do is I'm going to click on the retrieve button, and it lists the sequences here, and I can click here to get a better view, which will then allow me to compare each of them. So let's say I want to compare the chicken sequence with the horse sequence. So what I'll do is I'll deselect the other sequences, and notice that now I only have 2 remaining, and it's these 2 sequences. And what I'll do is I'm going to click on align this time, and this might take some time. It's running what's called an alignment program. And what it's going to do is going to take those 2 proteins, the amino acid sequences of those proteins, and they're going to line them up, and they did that just now. And basically it compares each amino acid and sees where they are similar and where they're different. So here you see these letters. This is the chick sequence on the top, and it continues here, and it continues here. Then on the bottom is the horse sequence, and it basically identifies the amino acids that are different and similar and identical. So I can go down here, and it'll actually give me the numbers that I'm interested in, and that's the identity. So you see that those 2 sequences are 67% identical to each other. And then I can record that information. Now let's say I want to go back to that original list and pick another 2 sequences to compare. So I'll just use my web browser back button. And let's say this time I want to compare the chickens. I'll keep that one with, the mouse. And then I'll go to align, and it'll run the alignment program again. And then I can keep on using my back button on my web browser. Let's say I want to get rid of one of the sequences. I can just put an X here, or if I want to get rid of all of them, clear. Now for the second part of the lab, we can go back to the home page. And this time I'm interested in hemoglobin of bats. Let's see. Let's search for it. It's going to give me a list of the hemoglobin beta proteins for different species of bat. And so what I can do now is, let's say I'm interested in this sequence. I've selected it. It's added here. And let's say I want to find out, are there other sequences that are similar or identical to this cave bat hemoglobin beta sequence? This time I'm going to press blast, and it's going to run the blast program, and that might take some time, because what it's doing, it's copying that sequence from that particular bat, and it's searching for that sequence throughout the database, all the species, to see if there are what we call hits, or similar sequences. And it give us the species name, as well as the percent identity. And we can sort of make some interesting conclusions based on this data. So it's given me my list, and it gives me the list in one form, but there's a detailed list down here. So of course the first entry is listed by percent identity. The first entry, of course, is going to be the cave bat, right? Because I used the cave bat sequence, so it's going to be identical to the cave bat. And then it's going to give me this list of other sequences that are identical to it. So if you notice, the second most similar is a bat, third most, another bat, another bat. But what's interesting, we get down here, and you see there are other species here, other types of organisms that are not bats. But if we keep on going down, you see, oh, there's another bat, and see if there are any other. Yep, another bat. And if we kept on going through the other pages, then we'll see other bats. So what's interesting here is that either that this is just by chance that these sequences of these other organisms are more similar to this bat than the other bats, or maybe we've stumbled upon an interesting issue with the classification of bats. Perhaps bats are not what we call monophyletic, um, and not a true classification group, because if it were, then we would see all of the bats listed here as being more identical to each other than to some other type of organism. But we actually don't see that in this case. So I think that's a sort of interesting observation, and perhaps we should check other proteins to see if we get the same results. Thank you.